Hi, welcome to OA Week 2025. This is the Marine Organism Response to Multiple Environmental Stresses session. We'll just wait a minute for everyone to come in from the waiting room and then we'll start the session soon. Okay, we'll get started. Welcome everyone. Thank you for joining us for OA Week 2025 on our final day. My name is Molly Phillips and I'm an OA Week coordinator. Please note that this session is being recorded. OA Week is presented by 4 sponsoring organizations. First, GOA-ON, the Global Ocean Acidification Observing Network. Second, NOAA, the United States National Oceanic Atmospheric Administration. Third, the IAEA OA-ICC, the International Atomic Energy Agency Ocean Acidification International Coordination Center. And last but not least, the IOC, the Intergovernmental Oceanographic Commission of the United States Educational, Scientific and Cultural Organization, UNESCO. GOA-ON was established in 2012 with just a handful of members. Since then, GOA-ON has grown immensely. It now has over 1,200 members from 110 countries. GOA-ON also consists of 11 regional hubs, which span across continents and oceanographic regions. We've heard from most of them throughout OA Week. If you're not a member yet, you can join GOA-ON today by visiting GOA-ON.org, and we'll put a link in the chat for you as well. OA Week debuted in 2020 and returned in 2021, when events and conferences were postponed due to COVID-19. Following the successful in-person symposium on Ocean in a High CO2 World in 2022, OA Week has become an annual event to maintain momentum around OA research and provide a virtual platform for the ocean acidification community to exchange their latest findings. We are thrilled to present a wide range of ocean acidification topics and speakers from around the world. A few words on housekeeping before we begin. During the presentation, all participants are in listen-only mode. You're welcome to type any questions in the question box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring incoming questions and we'll pose them to our speaker during the question and answer session, which will be at the very end of the session. And for the discussion, you can use the raise hand function in the toolbox in the bottom of your screen, and then we can call on you to ask the question directly if you do have your hand up during that question and answer. And with this, I would like to introduce you to your moderator for the session. Courtney Witkowski is an associate research scientist with the International Atomic Energy Agency's Ocean Acidification International Coordination Center. She supports the center's capacity building program, which trains scientists across the globe to study the impacts of ocean acidification and multiple stresses on marine species. Courtney also serves as one of the GOA-ON Secretariat members. Courtney, the floor is yours.
Thank you so much, Molly, for that introduction, and to our audience members for tuning in. This session is Marine Organism Response to Multiple Environmental Stressors. Environmental stressors rarely occur in isolation, making it crucial for us to understand how multiple stressors interact in order to accurately predict and protect marine organisms under a changing climate. And our goal for this session is to share knowledge from ongoing ocean acidification research on marine organisms, with a focus on how additional environmental stressors shape their responses. We unfortunately are having a slight change to our schedule, and 2 of our speakers became unavailable just this morning. So we will potentially just have one presentation, and after that we will have some time for a Q&A session and discussion, and hope to potentially pose some questions back to the audience about your experience with multiple stressor research. So with that, I would like to start things off by introducing our speaker, Dr. Emma Pontes. Emma is a postdoctoral researcher at the University of Miami's Cooperative Institute for Marine and Atmospheric Studies, also known as CMAS. Her research focuses on the physiological and metabolic impacts of climate change stressors, such as elevated temperatures, ocean acidification, and hypoxia on reef-building corals. She is particularly interested in how the relatively understudied threat of hypoxia interacts with these other major coral reef stressors. Dr. Pontes is also an experienced seagoing scientist, having participated in research cruises around the world, including to Antarctica, Brazil, Spain, and Easter Island, to monitor long-term changes in ocean chemistry. And with that, I will hand things over to you, Emma.
Awesome. Thank you so much for that introduction, Courtney. Let me go ahead and share my screen. Okay. Can everyone see my screen? Is that good?
Yes, looks great.
Awesome. Cool. Thank you. So good morning. Like Courtney mentioned, my name is Emma Pontes. I'm a postdoc working with the University of Miami Cooperative Institute for Marine and Atmospheric Studies. And I'm very excited to be presenting my research in an experiment that we conducted over the summer, and a talk that I'm calling today, The Physiological Impact of Ocean Acidification and Hypoxia on a local South Florida coral species called Orbicella faveolata, also known as OFAV, as you'll hear me refer to it in this presentation. And the common name here is the mountainous star coral, if folks are familiar with that species. Very common Caribbean coral that we see a lot here down in South Florida. So speaking of corals, I'm sure we're all familiar that coral reefs are not only beautiful, but super valuable and important to coastlines, especially here in Miami and in South Florida. They provide not only jobs, they support the economy. They provide us with the majority of the seafood that we eat down here. And they're just inherently beautiful and very cool organisms. And again, I'm sure we're all aware that these beautiful and valuable reefs are facing severe threats. So due to climate change, coral reefs are experiencing things like elevated temperature, ocean deoxygenation, and these stressors never occur in isolation, right? So anytime we have these heat stress events, they're also generally accompanied by low pH and or low oxygen. And so there's this real need for these multi-stressor studies that simulate these real world scenarios, because again, when we do a lab study, we want to look at multiple stressors in combination as opposed to just single stressors, so that we're simulating these real world scenarios as much as possible. And like I said, I'm sure we're all familiar with the idea that corals are experiencing elevated temperatures, these heat wave events that can lead to mass mortality, ocean deoxygenation, and these localized hypoxia events can also lead to mortality. Obviously, the focus of OA Week here today, this reduced pH in our oceans, and also like things like pollution and eutrophication, nutrient input, Sorry, let me make sure. Oh, and pollution, like I said earlier, things like eutrophication and runoff and stuff like that. So coral reefs are facing many stressors, and these stressors, like I said, rarely occur in isolation. So it's really important that we study these together. Sorry, did I lose my slides? Okay. Sorry, I'm still like navigating, making sure I'm good that I can see what I'm supposed to be saying. Sorry.
It's okay. We can still see them.
Okay, perfect. So enter Fresca, essentially. So Fresca is a multi-pronged initiative that stands for the Florida Regional Ecosystem Stressors Collaborative Assessment. And the goal here is to examine the effects of these multiple environmental stressors on several different benthic ecosystems. So this is a really important initiative that involves several institutions that are all working towards kind of disentangling how multiple stressors work in combination. And there are kind of 5 key stressors that Fresca is interested in, and those include ocean acidification, hypoxia, ocean warming, harmful algal blooms, and eutrophication. And for the purposes of today's presentation, the experiments that I'm going to be talking about are focused on ocean acidification and hypoxia. So the way Fresca is organized is it's separated into these 4 different modules that each kind of feed into each other and utilize data from the other modules. So in module 1, we are looking at these biogeochemical models where we do a lot of kind of local research cruises where we can measure the biogeochemistry of South Florida and take that information and kind of put it into a model that we can essentially parameterize these biogeochemical responses in South Florida. So module 1 is kind of focused on like modeling based on real world measurements of carbonate chemistry parameters. Module 2 is where I come in and where the kind of lab-based experiments come in, where we are really interested in looking at the ocean acidification and hypoxia thresholds of several different benthic taxa. And we have some other experiments that plan to incorporate temperature as well. In module 3, again, this is another kind of modeling module where these reef habitat persistence models are being generated, and they utilize data from module 1 and module 2. So we can take real world data of our biogeochemical parameters, real world data of how these organisms do in lab-based experiments, and put all of that into a model that can essentially predict what reefs will look like under a changing climate and under these different combinations of stressors. And then in module 4, we also incorporate things like plankton and fish distribution and habitat modeling there. So we can kind of generally encompass all of these really important South Florida habitats and collect as much environmental data as possible to inform these models that we're looking at. And yeah, like I said, I'm kind of focused on module 2 here, where we have our lab-based experiments. So getting into module 2 a little bit more, we are, our main goal here is to develop these threshold profiles so that we can kind of understand how these different habitat altering species perform under different combinations of ocean acidification, deoxygenation, temperature, and nutrient levels. And at this point, we're still relatively early on in our experiments. So these experiments have been conducted on a couple different coral species, but eventually we plan to expand these experiments to analyzing not only corals, but also bio-eroding organisms like sponges. We also have plans to do these experiments on photosynthetic organisms like seagrasses and sargassum. And then we have partners at the Virginia Institute of Marine Science that are looking at how harmful algal blooms respond to these different combinations of stressors. So all of these experiments, for the most part, are conducted in the Experimental Reef Lab, which is really cool, newly renovated facility that exists at the University of Miami down here in South Florida on Key Biscayne. The Experimental Reef Lab consists of 16 different independently controlled aquariums. Each aquarium is equipped with a thermometer, pH sensor, and oxygen sensor so that we are able to manipulate the pH, oxygen, and temperature conditions of each individual aquarium. And we can kind of, people have actually referred to this as like climate change in a box, which I think is pretty cool, where we can set these very distinct combinations of pH and oxygen levels that are completely independent of one another from separate tanks. So we can get this really nice high replication, high resolution level of multi-stressor experiments in this one room. So now I'd like to kind of walk you through our experimental timeline and kind of our experimental design. So our experiment started in May of this year, where we started with some baseline data collection. We measured a bunch of different physiological parameters to kind of start as our baseline before we started with any manipulation of pH or oxygen. Following that initial baseline data collection, we then started up with 4 different pH levels ranging from 7.7 to 8.1. So these represent kind of pre-industrial, contemporary pH levels down to, projected pH levels that we expect to occur by the end of the century and into the future in terms of, our kind of most severe ocean acidification level. And so we maintained that those 4 different pH levels for 33 days. And all the while we were maintaining our oxygen concentration at just ambient oxygen. So that comes in at about 200 micromoles per liter. And per treatment, we had 8 individuals, 8 Orbicella faveolata fragments per genotype. And we wound up having 3 different genotypes. So we were able to look at different genotypic variability among responses, as well as longer term, we plan to look at species-specific responses. But yeah, we essentially had 8 organisms per genotype per treatment. And so that single stressor phase occurred for 33 days. After that single stressor phase, we then reached our second time point of data collection, where we again performed all of these physiological measurements to understand how the corals are doing under this ocean acidification, under these OA conditions only. And I wanted to quickly show what our tank conditions kind of look like for that OA only phase. And I realize this is like super messy and there's a lot of colors and squiggles going on here. But basically the point here is that within our 8 different aquariums, we are able to, if we look at pH on the top here, oxygen on the bottom, you can see our 4 different pH levels parse themselves out really nicely. The other thing that's really cool about the experimental reef lab and what we were able to do in our experiments is to simulate this dial oscillation in pH. So every hour of the day, we have these different pH set points where we can mimic how pH fluctuates daily on a reef. And again, the whole goal of these experiments is to simulate real world conditions as much as possible. So we wanted to include that dial oscillation of pH into our ocean acidification kind of studies. And similarly with oxygen, we're seeing, this range of oxygen all the way down from, about 170 micromoles per liter, all the way up to sometimes, 240 micromoles per liter. And this is pretty standard daily dial fluctuations of oxygen that we would see on a normal reef. But again, those averages and the target set point are those targets that I mentioned in the previous slide. So coming back to our kind of schematic for the timeline and our experimental design, after that OA only collection, data collection time point, we then started our multi-stressor phase, where we maintained the same pH levels, but now we split up that treatment into a mild hypoxia treatment and a extreme hypoxia treatment. These mild and extreme hypoxia values are based on literature values. The kind of literature definition of hypoxia is about 62 micromoles per liter. And then our mild hypoxia is just below that minimum that we see in our daily oscillation. So these are taken from real world situations where we are kind of mimicking this extreme hypoxia situation and our mild hypoxia situation. We then carried out that multi-stressor phase for another 26 days. And then at the end of that phase, we did another round of data collection. So we have these kind of 3 main time points where we can look at our baseline, our single stressor phase, our ocean acidification only, and then our multi-stressor phase where we can look at ocean acidification and hypoxia in these 2 different severities at the end of our experiment. And then looking at the data here for our multi-stressor phase only, we did encounter a couple challenges in our experiment where, this system is relatively new and we are still kind of like working out the kinks of how to really precisely control pH and also manipulate oxygen at the same time, which is, as it turns out, pretty difficult to do. So what we decided to do for the multi-stressor phase was actually get rid of the diol cycling, and that made it actually a lot easier to control our oxygen. So that was one of the challenges that we faced, but so that's why you're seeing for our pH levels, we wound up just going with static pH here instead of that daily oscillation in pH that we had in the first phase. And then in terms of our oxygen, we decided to go with what we're calling like an intermittent exposure to hypoxia. And again, this is a pretty good simulation of what occurs in the real world. These hypoxia and localized mortality events due to low oxygen tend to be pretty short-lived and intermittent. So we decided to mimic that as best we could with these kind of so the red boxes are essentially showing where we have our low oxygen phases. That middle line represents our moderate or mild hypoxia treatment, and the lower levels represent our extreme hypoxia. And again, we were essentially trying to mimic this intermittent exposure to hypoxia here. So in terms of the data that we collected and how we are kind of assessing how the corals did under these conditions, we measured a lot of different things. We looked at coral growth rate by measuring the buoyant weight of the coral. We looked at the photosynthetic efficiency by performing IPAM. We can also measure their calcification rate by looking at the total alkalinity anomaly during incubations. We also performed respirometry incubations where we can measure the animal respiration rate and the photosynthetic rate of the zooxanthellae. We took photos of the corals at every time point so we can measure their paling and kind of bleaching response. We also looked at linear extension to measure their vertical extension rate. And actually this linear extension we only performed on the branching coral species that we did this experiment on. We also were able to 3D scan our corals so that we can measure their surface area. And a lot of these values are normalized to the surface area of our corals. And then finally, we're just performing regular health and mortality monitoring so that we can eventually generate our response curves. So looking at our first set of data, oh, and I forgot to mention too that the data that I'll be presenting today, I unfortunately don't have time to share all of this data, and we're still in the process of working up a lot of this data, but what I want to share today is our respiration incubation data and the IPAM data. So first, looking at the dark respiration data, these incubations were performed in the dark, obviously. Corals were dark adapted for about an hour, and then we can essentially measure how much oxygen they consumed in the dark in these kind of closed incubation chambers. So first, this panel on the left is our first time point, that initial kind of baseline data. And just under ambient pH and ambient oxygen conditions. And the respiration here is very much in line with other studies that measure respiration in Orbicella faveolata under these kind of ambient conditions. Then in this next panel, I'm showing our second time point, which represents our normoxia phase, normal oxygen conditions, but now we can look at our 4 different pH levels. And what's interesting here is that the data kind of presents itself in a bell curve. And this is generally what we expect to see, where we have, our highest rates of respiration at that 8.0 pH. With our lower pHs generally having lower respiration, which again is a sign of stress. When these corals are stressed out under these lower pH levels, they're not able to respire at the same level that they would under ambient and kind of comfortable conditions. Then we can look at our other 2 panels that represent our third time point of data collection, where we have our moderate hypoxia conditions under 125 micromoles per liter, and then our very severe hypoxia under 62 micromoles per liter. And again, we still have those 4 pH levels parsed out in our 2 different hypoxia levels. And this is where I think the data gets really interesting. And let me also go ahead and add our statistics here. And again, I know this is like a lot of letters and things to kind of tease apart. And I've spent a lot of time looking at this data and trying to understand like what is the kind of take home message here. And one of the things I want to point out is our 7.7 treatment. So in kind of our ambient conditions of 7.7, the difference between, that 7.7 treatment at ambient versus even moderate and severe hypoxia is significantly different. So this addition of hypoxia significantly reduced respiration, which again is just pointing to this idea that when we're looking at, when we want to understand how corals respond to stressors, it's really important to consider multiple stressors, because we're seeing a significant reduction between, our ambient conditions in terms of hypoxia compared to now when we add hypoxia into the mix with ocean acidification, we see that significant reduction. The other interesting thing here too is that it doesn't seem to, like the magnitude of hypoxia doesn't seem to have a significant effect on our respiration right here. Any addition of hypoxia is significantly reducing respiration under this 7.7 treatment. And you can also see that in general, our lowest hypoxia treatment also just overall had the lowest respiration rates, which again makes sense because obviously respiration is oxygen dependent. So as soon as we reach that kind of multiple stressor phase where we have severe hypoxia and this addition of ocean acidification, we're seeing these significant reductions in respiration. Another interesting point here too is that in our elevated pH, we're not seeing a significant reduction in respiration when we compare our moderate hypoxia to our ambient treatment. We really only see a significant reduction in respiration when we incorporate severe hypoxia. So this kind of points to the idea that elevated pH could almost alleviate the negative effect that we see from severe hypoxia when we're considering multi-stressors. So again, this just kind of driving home the point that multi-stressors kind of work synergistically, where they have this compounded negative effect compared to just single stressors. So now looking at the photosynthetic rate. Here, we performed these incubations in the light. So we're able to measure how much oxygen the coral is, or rather the zooxanthellae, that algal symbiont is producing in the light under these different combinations of stressors. So first, this data is structured the same way, where on the leftmost panel, we have our first time point of our photosynthesis under just kind of normal ambient conditions. Then we can introduce our second time point, which includes these 4 different stressors, or rather 4 different pH levels here. The one thing I want to point out too is that these error bars, there's a relatively large variation in responses, and this is actually due to those different genotypic responses, which I unfortunately didn't have time to put together some figures to show those kind of different genotypic responses, but we did see relatively large genotypic variation in responses, which is another kind of interesting point that even within the same species, different genotypes respond very differently. But we are seeing similar kind of bell curve shape with our second time point. And then we can also look at our final time point where we are incorporating that mild and severe hypoxia. And I'll go ahead and add the stats there. This data is a little messier than the respiration data, but still some kind of interesting patterns here. And so in general, hypoxia, this addition of hypoxia is generally lowering our photosynthesis and respiration rate. And what I wanted to highlight here is if we look at our 7.8 treatment, which just kind of progressively gets reduced under moderate and then under severe hypoxia, we just continue to see a decline in photosynthesis. And this was relatively surprising for me because hypoxia is not, or rather photosynthesis is not an oxygen dependent process. So, this points to the idea that hypoxia is stressing the coral out and potentially, damaging the photosynthetic machinery that the coral uses to perform photosynthesis. That was one of our hypotheses. But in general, we are seeing these reductions in photosynthesis once we add that additional stressor of hypoxia. Again, just driving home the point that this multi-stressor response is actually very different than the single stressor response. And finally, looking at the IPAM data, which again is a measure of photosynthetic efficiency, there were no significant differences here, which kind of disproves my hypothesis that I just mentioned on the previous slide, that the stress of hypoxia is actually damaging the photosynthetic machinery. These data prove that that's not the case, that there is still that machinery, that ability to perform photosynthesis is still maintained. So again, we're not seeing any significant differences here across time point, across treatment. And my thought here is that, the IPAM measurements are essentially this really quick snapshot measurement of the maximum quantum yield, right? Which is kind of a theoretical best case scenario, how are the corals able to perform photosynthesis? So what this data to me is saying is that, in the snapshot measurement, the coral photosynthetic machinery is still intact and is still able to perform photosynthesis to its best ability. But when we do these longer incubations, we're seeing that some portion of the coral is overwhelmed and is not actually able to perform photosynthesis at its highest level that it would be able to under ambient conditions. So in conclusion, we found that both respiration and photosynthesis were significantly reduced under these multi-stressors, and that it seems that oxygen concentration is really the main driver here for responses as opposed to pH, but that combination of pH and oxygen can elicit really different results, and again, highlighting the importance of these multi-stressor studies here. And yeah, my next bullet point, exactly as I just mentioned, the single versus multi-stressor effects are significant. Just highlighting how important these multi-stressor studies are, and that, studies that have come out that are analyzing only single stressors, may be missing some portion of coral response, especially when we're considering, informing reef persistence models and considering how corals are going to fare in the future. It's really important that we are considering the whole picture and looking at these multi-stressor measurements in combination. And then finally, I was really interested in this result of the IPAM versus the metabolic measurements, and the fact that we saw no significant difference in IPAM, whereas we did see significant reductions in our photosynthesis rate. And again, I think that comes down to the IPAM being this kind of snapshot, idealized measurement of coral photosynthetic efficiency, whereas the photosynthesis incubations are kind of longer term, real measurements of how the coral is performing in that actual treatment. And with that, I'll be happy to take any questions. Thank you.
Thank you so much, Emma. So we will have some time for questions, and I encourage people to put any questions they have for Emma in the Q&A box. But we are actually going to do a slight pivot before we go to some questions for Emma. And we have a short poll for the audience. So sorry, just one moment, and I will share my screen because we would like to engage all of you who took the time to attend this session today. So we just have a few short questions to learn from the audience what type of multiple stressor research you are engaged in. So if you scan this QR code, or my colleagues are putting a link in the chat that you can use to join. So I hope that people are able to pull this up on your phone, their computer. And we will have just a few short questions for people to answer, just so we can get kind of a snapshot of what type of research you all are engaged in if you are doing multiple stressor research. Okay, and my colleague Molly is helping out with this, so I think I will maybe stop sharing so that she can start to share her screen as people answer. But our first question for people to answer will be, what stressors are you researching? And I'll maybe add, if you're not engaged in multiple stressor research, but something that you are interested in undertaking in your region, maybe answer what stressors would be most important for you to consider researching. So you can select as many as would be applicable to your studies. I will maybe just wait one more minute or so for people to answer. And yes, Molly, I can see your screen. Thank you for sharing that. So it looks like we have a few people, of course, looking at carbonate chemistry parameters, one on oxygen, one on metals, and one on plastics. So a little bit of a range there, which is interesting. Let's maybe move on to the next question, if people have started to. Okay, so now this is open for people to answer. So which organisms are you looking at the effects of multiple stressors? We just heard a great example from corals, but of course we know that this type of research can be applied to others. So would be curious to hear from people what type of organisms you are researching through your work. Or again, what you are interested in researching if you're not already. Great, thank you for those who have contributed responses. Really a good range here. So from jellyfish, macroalgae, sea urchins, copepods, sharks. Looks like maybe someone else is researching corals with you, Emma. Okay, and let's go on to our last question. And I guess just to foreshadow, Emma, I know this is something you touched on in your presentation a little, but I think it could be interesting for us to talk a little bit more during the discussion. But we would love to hear from the audience. What barriers or challenges do you face in conducting research that considers multiple stressors? Two similar answers to the first question are technical constraints and expensive equipment, limited budget and experience, so maybe a need for collaboration, the volume of tests that are required to determine the interactions between a range of stressors. Great. And then requires more work to understand and characterize natural environmental regimes. Yeah, I think we heard Emma referencing how you had to consider the environment that your corals would be facing and ambient conditions when designing your experiment, and very similarly, missing baselines and obtaining gases in remote locations. So a similar technical equipment limitation. Great, thank you everyone for submitting those responses. I think I'll give it one more minute just to see if we get one or two more stressor dichotomy. Since responses are slowing down a little, maybe Molly, we can, take that down. Thank you everyone for participating. I think it's interesting for us just as we move into a few questions, just to know who is in the audience, at least at a high level, and what type of research you all are interested in or undertaking. And so, Emma, I think maybe just to start with a first question, I think I would be curious to hear you expand maybe on the challenges of doing multiple stressor research. And I think we, heard, I think one great tangible example from you was the controlling of the pH and oxygen and how you had to make an adjustment. But I'm wondering if there were any other challenges that came up.
For sure. Yeah. So, like I said, the experimental reef lab is newly renovated. So actually, this Orbicella faveolata experiment was the first experiment that we conducted in this system. So it was kind of a learning opportunity for us kind of along the way. We were, we prepped as much as we could, but yeah, we obviously ran into challenges, and we're trying to address them as we went. But one of the main things is figuring out the best way to control pH and oxygen at the same time. Our systems have a gas delivery method. So essentially each aquarium is equipped with a module that consists of 3 different mass flow controllers, which all dispense 3 different gases. So we're working with air to raise pH and raise oxygen, carbon dioxide to lower pH, and nitrogen to lower oxygen. So there was a lot that went into what's called the PID algorithm that determines how much gas to dispense and when to reach our target values without overshooting. And then, we're constantly working against environmental conditions. Like oxygen wants to raise, right? Like we're constantly fighting maintaining that low oxygen level, and then throw in trying to control pH and hypoxia at the same time was very, very difficult. And what we found, one of our main challenges, and part of the reason why we decided to go with this intermittent hypoxia, is because we were going through nitrogen at an insane rate. Like we were going through 300 cubic foot, a pretty standard size cylinder, 2,500 PSI cylinder of nitrogen, one of those cylinders every less than 24 hours. For like 3 weeks, which was intense and not sustainable, and gets very expensive. So we were having to come in every single day, coming in on the weekends. We even set up like a nanny cam, essentially, that we could log into remotely, so we could see what the PSI of the tank was at all times. So if someone needed to come in and change the tank overnight or whatever, we could do that. So really that combination of maintaining multiple stressors in one system was really, really tricky. And the solution that we found to that was the way that we are delivering gases to our tanks. So we initially started with an air stone that was bubbling in the sump, and then it's kind of like a recirculating system where water is going from the sump into the aquarium. We decided against the air stone and switched over to using a pump with a venturi, which is essentially a pump that has an attachment that breaks up the bubbles into just much smaller bubbles. And so we found that the smaller the bubbles, the better, the more efficient the gases are, which makes sense, right? Like the smaller the bubble, you have higher surface area to volume ratio. So the bubbles are able to interact with the water better. They stay in the solution longer. So it's actually more effective. And we were able to reduce our nitrogen consumption by like 70%. So we're actually in the middle of running an experiment now with our new system, and it's working really well. But yeah, it's really, really tricky when you're delivering all these different gases to maintain. And that's why we decided it was just too much to do the diel oscillation in combination with trying to reduce oxygen. So yeah, if anyone else is dealing with that, I totally understand. It's really, really tricky.
Yeah, I think that's really interesting to hear about some of the details of the challenges you faced and just how those solutions came about, especially when we're we're hearing, I think, calls for more multiple stressor research throughout other sessions at OA Week, which I agree is really important, but I think it is also critical for us to take the time to hear about the real challenges and what it takes to address those. So thank you for sharing that example. And so my next question, I wanted to go to an audience question that we received. Verena asked if you also measured p-crit in your experiment, and then a follow-up was, did you find similar responses in other coral species?
Yeah, thank you so much for this question, Verena. Maybe you've read my other research. I have a paper that measures PO2 crit under different oxygen concentrations. So I love this question. Awesome. Okay, cool. I'm really psyched that people are reading that. That was kind of like my bread and butter in my PhD, and something that I was really interested and really proud of when I came out of my PhD. That was one of my favorite experiments that I did. And unfortunately, to answer the short answer to the question is no, we didn't measure PO2 crit here. To measure PO2 crit, we need a wider range of oxygen concentrations. And, the grant that I'm part of, Fresca, is really only interested in these two kind of like mild and severe hypoxia. So we weren't really able to get this like really wide range of oxygen concentration measurements that we would need to calculate PO2 crit. But it's something, like I said, it was one of my favorite experiments in my PhD, and it's something that I'm going to try to squeeze in at some point because I think it's really interesting. And I'm just super curious. So in that paper where I measured PO2 crit, we did include Orbicella faveolata. So it'd be really interesting to do that same experiment in addition to ocean acidification, and to look at different genotypes of OFAV and see if they have similar PO2 crits. So that's kind of like my passion project that maybe I can squeeze in at some point, but thank you for that question. And then, did we find similar responses in other coral species? Not yet. So we had, like I said, this O5 experiment is the first experiment that we successfully completed in this new system. We did do a very brief trial run with Acropora cervicornis, and this is back when we were still troubleshooting how our pH probes functioned. So we wound up having to end that experiment a little early, so I didn't get a ton of results, but I would imagine that different species are going to respond very differently. And there's other research that shows, I cite a lot of research by Maggie Johnson's group. They do a lot of deoxygenation research on OFAV and Acer and corals like that. And what they have found is that OFAV tends to be more resistant to just better at handling hypoxia and low pH. So I suspect that other coral species, especially more sensitive coral species like Acropora cervicornis, are going to be more sensitive. We're currently in the middle of an experiment right now where we're doing 2 species at a time, same experimental design, but the species we're working with are Psittacia siderea and Stephanosinia intercepta. And those 2 species also tend to be very resilient and resistant to climate change. So I suspect that maybe they'll be more resistant. Like I said, we're in the middle of the experiment, so I don't have all of the results there yet, but I would imagine that different species are going to have wildly different responses for sure.
Yeah, I see Verena also shared just now that she has done some OA and hypoxia experiments where they found very different responses between resistant and sensitive species, not yet published. So we'll definitely look out for that work. Great.
Very cool. Yeah, I'd love to see when it comes out.
Yeah, I want to go to another audience question we have, Emma, from Chandra Sakara. They share, Emma, your lab results on coral responses to multiple stressors are fascinating. From your perspective, how could these experimental insights be embedded into conservation policies that support reef restoration and community resilience?
That's a great question, and that is the whole goal of Fresca and of these experiments. So within Fresca, we work with a group called MTAG that are essentially managers, policymakers, people who are, stakeholders, people who are on the ground and making these policy decisions and implementing mitigation strategies to protect and preserve coral reefs. So the goal with these experiments is to feed this data into these reef persistence models. So my colleague, Dr. John Morris, is a postdoc within my group. He is the lead for module 3, I think it is, that does these habitat persistence models. He and I work together so that I can provide him all of my physiological data under these combinations of pH and oxygen stressors. So I can say, this is the growth rate of this coral species under this combination of stressors. And then Dr. Morris can incorporate that information into his reef persistence models and literally predict what reefs are going to look like under these different conditions. So if we can identify these kind of thresholds, sublethal thresholds and mortality thresholds for different coral species, we can, use that information down the pipeline to inform these reef persistence models that managers can then use to set caps on, past this point, this is what we expect reefs to look like, and this is how we need to manage reefs given this combination of stressors. So yeah, that's a great question, and that's the entire goal, is to basically inform these reef persistence models so we can better predict and conserve our reefs.
Yeah, that's super interesting to hear about how the different elements of Fresca connect for that end goal of communicating to the managers of these coral reef ecosystems. I have a follow-up question that I think it's a little related, but I'm wondering if you have come across there being extra complexity when communicating research results that include responses to multiple stressors, or maybe just how that affects the communication. Does it make it easier or harder for people to understand the importance of taking action? And I think I'm asking this, through the lens of we're in Ocean Acidification Week, so we are generally raising awareness about ocean acidification, but of course, your work is in this greater context of climate change and anthropogenic threats.
Yeah, for sure. It definitely makes it more complicated. As soon as you're introducing another stressor, the results get messier, and, you saw the results. It's not like a super clean story of, add this much hypoxia to your ocean acidification, and this is the response. Sometimes things aren't what we expect. I mean, I totally expected there to be a significant effect on the photosynthetic efficiency, and we didn't see any effect on that. So it definitely makes things more complicated, but that just means that it takes more time as a researcher to figure out what the story is and figure out what the best way to communicate that is, so that it can be this actionable data, because there's no point in doing these experiments unless we can actually inform management and work towards protecting our reefs. So it's, just an extra kind of lift on the researcher to tease things apart. Definitely involves some more advanced statistics, I would say. So, I've had to beef up my statistics background and figuring out the best way to tell this story that makes sense to managers and scientists. Definitely makes it more complicated, but I think is certainly needed for sure. But yeah, just kind of a heavier lift on the researcher in terms of statistics and communication. But all with the goal of trying to simulate the real world environment as much as possible so that we can inform and predict what reefs will look like in the future.
Yeah, it does seem like it's a more powerful message in the end when we can give a more comprehensive story about what is actually happening in this environment across different dimensions, but definitely some challenges and bringing people along on that story so that they can accurately understand what's happening. And maybe just one last small question. So part of the reason that we do these sessions at OA Week is to, of course, share new research results, but also build connections across different regions when we have attendees from all over the globe tuning in. And so I'm wondering if collaboration, either regionally or interregionally, has played a role in your work, or if you maybe if you can speak to a need you see for that collaboration to strengthen multiple stressor research going forward.
For sure. Yeah, I think that's a really important area of this multi-stressor research, mostly because there are massive challenges and massive roadblocks. And, I think if we had collaborators that had performed similar experiments, it would be very easy to reach out to them and be like, hey, what do we do? Like, we're running into this problem, and, how have you guys tried to solve this? And just have some kind of like sounding board to like troubleshoot and problem solve. And we don't really have that currently. We are, within Fresca, we have a lot of different partners that are different institutions, different national and federal organizations. But within my module and these experiments, these are really the only experiments or lab-based portion of Fresca. So I think that's definitely something that would be super helpful, just to have collaborators, even to just commiserate with, and just to, kind of get help from. And, that being said, I'm remembering too that, when we were having some challenges with understanding how the best practices of working with the pH probes that we have. And we had reached out to a couple scientists at Embari in California that utilize and have published on these pH probes. They are Pyroscience brand. They're Pico pH sub probes. So if anyone's working with those and has questions or has insight, I'd love to hear that. But yeah, we reached out to them, and they provided us with a lot of really helpful stuff that helped us kind of get off the ground and make sure that we were calibrating the probes as much as we needed to, and making sure they were accurate and all that kind of stuff. So absolutely, collaboration is super important, and we are certainly open to working with people, helping people get their own experiments up off the ground, especially since we've done a lot of the legwork of like figuring out how to make this work and going through some of those challenges already. So yeah, we're definitely open to collaboration in that sense, and would be super happy to engage in that.
Yeah, that's great to hear. I think GOA-ON has been really successful in building some of those collaborations and connecting people even across different regions in the past, and something that we are definitely aiming to continue to foster going forward. And even, I guess, more personally, my organization, the Ocean Acidification International Coordination Center at the IAEA, is also specifically focused on capacity building for multiple stressor research, as Molly mentioned at the beginning. And part of that is building this network of researchers who are working on that topic and connecting them with some of the experts that have been leaders on this topic. And so thank you again, Emma, for the great presentation, the thoughtful discussion. But with that, we're nearing the top of the hour. So I think I am going. Sorry, muted early. I'm going to turn it back over to Molly to help close us out.
Thank you so much, Courtney. And thank you to Emma for giving us this talk today, and thank you the audience as well for joining in and contributing to the discussion around multiple stressor research. And also for joining us at OA Week 2025. We have a few more sessions, 2 more sessions of OA Week this evening, if you want to, well, this evening for me, the rest of the day for wherever you are. So do register for those if you're interested. And if you've missed any of the sessions so far, you can catch up with them on our OA GO-ON YouTube channel. If you would like to stay up to date with the GO-ON community, then consider signing up to be a GO-ON member, which you can do through the QR code on the slide here. Thank you all very much, and see you maybe later today.
Thanks, everyone.